Hi, my name is April Kriebel, and I was part of a research experience for undergraduates at Auburn University this summer, and we explored transcription factor binding site diversity in Drosophila, and I worked under my mentor, Dr. Rita Grays of Grays Laboratory. So first of all, why Drosophila as a model organism? They have large populations that can be maintained with relative ease, both physically and spatially, which makes them a good model organism. They also have a high number of offspring, which makes them prime for genetic studies. Furthermore, they have a short development span, meaning that longitudinally there's not a lot of temporal difficulties in doing studies. And they're also well conserved to the human genome. Although they only have 4 chromosomes to the human 23, they still have a lot of similarities that we can take what we've learned in the fruit fly and apply it to human genetics. So let's talk about why we wanted to assess transcription factor binding sites. Now, when you have a gene, you have the part that's expressed, and then you have regulatory regions which transcription factor binding sites will bind to. Now, these regulatory regions help either heighten or lessen the expression of a gene, but often they don't turn it completely on or off, which makes assessing their effect on the organism difficult. And so we wanted to look into that. And specifically, we wanted to look at FOXO and endoceno signaling receptor pathways, both of which are transcription factor binding sites. And these have both been linked to secondary sex characteristics in Drosophila. Now, what is a secondary sex characteristic? In order to understand that, you have to understand how Drosophila's sex are determined. They function through chromosome counting mechanism, which means that the way their sex is determined is they have a mechanism that looks at their chromosomes and counts the number of X's. And so it's not determined by a double X or an XY, merely the number of X's present in the organism. The number of X's will then either turn off or on the gene called sex lethal. Now, this sex lethal will go on to control what we know as the secondary sex characteristics, which are represented by external genitalia, physiology, and behavioral things such as mating rituals. So for our research objectives for the summer, the concentration of the REU was to use Temple, which is a software that I'll talk about later, to assess the difference in transcription factor binding sites between certain populations. What we want to eventually do with this research, as you can see through steps 2, 3, and 4, is we want to take the genomic analysis and identify signatures of adaptive evolution. And then we want to look for differences in the phenotypes as a result of those differences in the transcription factor binding sites. We'll then take that information and use it to evaluate whether those phenotypes are expressing sexual differentiation. So why do we want to search for transcription factor binding site variations among multiple populations? And to look at that, we need to understand Drosophila's origins. It started believably in Africa and has spread out throughout the globe since. So by assessing in different populations, we can see how it's evolved in different continents. So let's look at our data. The data we used was collected as part of the Drosophila Genome Nexus, which has over 498 genomes of Drosophila which have been collected globally. And what's unique about this is they used a consistent pipeline to gather all of this data, and so it's a very rich data bank that you don't have a lot of problems with statistically to analyze because it's all been analyzed through the same pipeline. And we specifically decided to focus on 3 populations, one in California, one in Ithaca, and one in Rwanda. Now, why did we pick these 3 populations specifically? Well, as I mentioned before, the Drosophila was believed to have originated in Africa, and so the one population from Rwanda functions as more of our control population where we're assessing the difference between those and as it's spread out. Now, we wanted to pick one in California and one in Ithaca so we could assess both the difference between those 2 populations and back to the home population in Rwanda and see how the Drosophila has changed. So what methods did we use in our research? We used a software called Temple, which is transcription elements mapping at the population level. Now, what's unique and novel about Temple is that it's comparing the population's divergence, but it does it while accounting for within population divergence. No software has previously done the within population divergence part. And the way it does this is it uses position weight matrices to predict changes in binding affinity. Now, if you look at these, the bigger letters represent nucleotides that are more likely to be present, and with the smaller nucleotides, those are ones that are either shared or not as likely to be expressed. And so it compares the differences between the PWM matrices and what's actually found when you sequence the genome to give you your results. Now, the way it compared those, it used QST and FST. Now, QST is assessing the variance between populations for the trait's PWM score. And then the FST is the difference between population at a specific SNP loci, which is a single nucleotide polymorphism. So that's how it was assessing the changes in binding affinity. Now, let's look at our results. So the results pictured here are simply for the X chromosome, but our research centered on